As a quick review from where we last left off, you talked about how the different types of, and I wanted you to be able to use the logic on the different types of amino acid properties and with respect to how they interact, the proteins that have them interact with the membrane, because this becomes important later on. And in particular, we discussed how it's we discussed how it's primarily hydrophobic regions in the interior, and that if there are any polar interactions, those are very important for protein-protein interactions within the interior, kind of holding it together. Now, we tend to have polar side chains on the two sides, since both the cytosol and the extracellular matrix will be aqueous or polar environments. And then many times there's this hydrophobic, in particular it's a aromatic polar tyrosine and tryptophan-like cap that goes right alongside the head groups of the phosphate bilayer. Then I just wanted to show you that not all of the integral membrane proteins are transmembrane alpha helices. There are several that do have beta sheets in them, and these typically, many of these tend to be, pores or receptors in that sense. And so many of them form what's called beta barrels. So if you remember from, chapter 3 or 4, many chapters ago, a beta sheet is not perfectly flat. There's a slight torque or twist to it. And so if you get a lot of beta strands together, it does create almost like an inner tube look. And so that's why it can be a beta barrel. You can also have what's called an alpha beta barrel, where it's a mixture of both alpha helices and beta sheets. And so these are just some of the examples of the pore complexes and things like that that can form within the membrane of the cell or even the organelles. And usually, it's very important for whatever their functionality is that they have their respective shapes. So I would just expect you to be able to discuss some examples, or if you're given a structure, you can say, oh, this is a beta sheet or beta barrel, or so on and so forth. You do not need to memorize all of the possible shapes and secondary structures and motifs and all that kind of things. And don't memorize all the examples that they give here, because it can go on and on and on. There's one called the Tim barrel, which is huge, and it's a common motif as well. Okay. And we've also discussed the different types of lipid anchors. For example, with the type 5 proteins. And so some of these we've already talked about in great detail. So for example, the farnesyl or geranyl group. Farnesyl means how many carbons are in a farnesyl group? Farnesyl means 15, whereas geranyl is 20, because each geranyl is a 10. Okay. But these are just really large isoprene units, and they are always held by a cysteine. Okay, so it's a cysteine on the protein that gets farnesylated or geranylated. And so one example of that we talked a lot about was RAS, but RAS is not the only protein that gets these type of lipid anchors added onto it. But you can also have fatty acids. So palmitoyl and myristoyl groups, those are just palmitic and myristic acids that are just added on. So there's long fatty acid side chains. And as they're showing here, it could be cysteine, serine, or even the amino end of an amino acid that can be lipidated. Okay. And then we also have really large complexes. This is a GPI. And so it has an inositol, which that's just a type of sugar, but it's a polysaccharide anchor that helps hold it into place, and so they can be quite complex. So now we're going to move on to membrane dynamics. What do you suppose that means? What does it mean when we talk about dynamics?
Changing.
Okay, changing. What else can it mean? With respects to what?
How they interact.
Maybe interactions. It could be even some things with kinetics or speed or different reactions that could occur. Okay, it just means that it's not static. Okay, things are constantly in the state of flux. And so we have to have it tightly regulated, and we'll talk about one or two specific examples of that and like what happens if it's not tightly regulated. And so there are a couple of different ways to think about the movement, just the physical movement of lipids. One is laterally or side to side. So this is very, very, very fast. Okay, and so they're just showing like 1 micron per second or something. I don't know how that it was going to depend upon the how crowded it is, like the components of what that lipid is. But it is very, very fast. And it's uncatalyzed. It doesn't require any type of enzyme to help the lipid move in that sea of lipids. Okay. So it is uncatalyzed. What do you suppose about trying to move it from the outer leaflet to the inner leaflet or the inner leaflet to the outer leaflet? Right, this is going to, why will it require something in order to get this dark colored phospholipid here to the other side of the membrane? Why would it require energy?
Because it's going through the hydrophilic.
You're going to have to take this hydrophilic head group and push it through the membrane, which is hydrophobic, in order to flip it out or flip it in. Okay. So you can have this trans bilayer movement, but it does require a lot of energy, okay, because if you don't catalyze it, as I mentioned, it's very, very slow. They're showing the T1/2 would be in matters of days, weeks, months, because it's almost impossible to get that phosphate to flip. It's not wanting to flip from the outside to the inside or the inside to the outside. Okay, and this trans bilayer diffusion, sometimes it's called flip-flopping. So it's not just in politics where we're talking about flip-flopping. Sometimes flip-flopping is good, but sometimes it's bad. That's not faith integration, it's political integration. Okay.
Are you allowed to do that?
Political integration? I would think so. Did I purport to go for one party or the other, even though they do tend to be a little one-sided here?
Just a bit.
Mm-hmm. And so, and if you don't, so there are enzymes that are used to catalyze this movement from one side to the other side, from the inner leaflet to the outer leaflet. And there are 3 classes of enzymes. There are the flip bases and the flop bases. These are my favorite named enzymes, I think, probably in all of biochemistry. Flip bases, flop base, and then the third one is even better. But I do expect you to know they are very specific. Okay, so flip ace and flop ace both require ATP because they're going against a gradient here. And so the flip ace goes from the outside to the inside. So it's the one that has the I in its name, which means it's the one that's putting the product on the inside. Whereas flop ace moves it to the outside, and it's got the O in its name. If you're going to go to medical school, you're going to have to learn how to do these mnemonics and little things like that. I still remember the 12 cranial nerves, well, the saying, I don't remember all their names, but the ooh, to touch and feel very good velvet, ah. And that was in our anatomy book, by the way, there, so I did not come up with that myself. But we always had mnemonics. But no, flip base and flop base. And that P-type versus ABC, those are specific types, and we'll learn more about the P-type ATPases next semester, but those are proton type ATPases. And it's not just, does anyone know what's another type of P-type ATPase? I mean, there's a whole class of drugs that's, now you can get them over the counter. You don't even have to get a prescription for it. That's the proton pump in your belly for those who have like acid reflux disease and things like that, GERD. And so Prilosec, omeprazole, Nexium, those are inhibitors for P-type ATPases. And that's why sometimes they have some really weird side effects for some people, because they, in theory at least, could potentially inhibit some other P-type ATPases besides the one that's in your belly. Well, I'm on a Mike Myers kick today. But I do want to point out the fact that this flip ace moves PE, which is phosphatidylethanolamine, and PS, phosphatidylserine, from the outside to the inside, and that's very important. That's the flip ace. And once again, these are going against the gradients, and they require ATP. And now, one of my other favorites, there's flip ace, flop ace, and there's scramble ace. One student kept calling it scramble eggs, but not eggs, ace, as in the enzyme. And it can go in either direction. It does not require ATP because it flows with the equilibrium, okay? And so it's the peacemaker of the, if I'm going to anthropomorphize it, it's the peacemaker of the trans bilayer enzymes. And it increases with respect to calcium, okay? And that can be due to a whole host of things, which we have talked a little bit about cell activation with respect to like muscle cells earlier with calcium, but those are not the only cells. So nerve impulses are calcium regulated. The pancreas, calcium plays a huge role in insulin regulation as well. You can also have cell injury, apoptosis, necrosis, things like that can also be can affect scramble aces. Okay, but it can go, once again, it does not require any ATP or any outside energy.
Lipids on
Right.
the outside, normally.
So, what more lipids? I mean, it's not going to have an. So, well, it's the identity. I don't know so much if it's, because there are different classes of phospholipids, but here, this is the reason why I really wanted to stress the fact that PS in particular, that's phosphatidylserine, is because that's the way that triggers apoptosis. Okay, and so the flip bases only move the aminophospholipid from the outer leaflet, and you have to keep this asymmetric because one of the important ways that what will become the apoptosome and the macrophage knows that a cell is in trouble is if you have phosphatidylserine on the outside. So if you have an increase or too much PS on the outside, then that signifies, okay, this cell is in trouble and it needs to be killed. So that's one of the reasons why this enzyme helps keep it from premature death, cell death, in that sense, okay? But phosphatidylserine itself, the enzymes that stick it to the outside or its expression to the outside is linked to a whole host of the apoptotic cascades, including those from, now I can't think of the caspases, and the release of cytochrome C from mitochondria, which we actually talk about in great detail next semester. So this is just, we want to keep some of this in mind for whenever we start to discuss some of the metabolic pathways. Since cell death is a very important part of life, Hakuna Matata, Disney integration.
I did not digest.
You did not digest, or I was going to say you did not inhale the brownies. You just slowly partook of the brownies. Okay, so microdomains, if you imagine the membrane as being like the sea of lipids, which I keep referring to it as, there are these regions that are highly specific where proteins come together and accumulate, or even certain lipids or carbohydrates will come together and they associate with each other. These little small regions of highly populous regions, for lack of a better term, those are called microdomains, okay? And many times they even call them rafts. So they really like that sea metaphor. I did not come up with that myself. I would like to take that point of notoriety, but I am not the one that can come up with that. So, but they do consider them like rafts because they do float, these islands float, so to speak, on that lipid membrane sea, since it is relatively fluid. And it's very important because many times the functionality doesn't occur. It's either inhibited or activated on whether or not it forms in these microdomains. And we're going to go for some specific examples in a moment, like for example, for endocytosis or exocytosis, for the fusion, the integrins, which will be next chapter, I do believe, for cell-cell signaling and things like that. Those require very specific complexes to be present. And so this is just a figure from your book, but you can actually see some of these. The one that, let me blow it up. The one that they're showing here is one that has to do with caveolin. And caveolin, in a moment, I'm going to show you how when you have a dimer or a multimer of this is what helps with the concavity of the membrane. But they associate with very specific lipids and carbohydrates and things like that. And you can actually see this in certain types of microscopy. And so that's why they're showing here an actual microscopy of this raft formation on the sea of lipids, so to speak. So we see a higher percentage of proteins and or specific lipids that kind of aggregate together versus the generic phospholipid region. Please ignore that. Hopefully that does not come through. And so I've already mentioned endocytosis and exocytosis, and these are just some of the examples of whenever we have to, or whenever we're talking about membrane interactions and membrane fusion in particular. And so endocytosis, of course, would mean, what is endocytosis?
Bacon.
You're taking it in from the external environment versus exocytosis, spitting it out, and that can come from the Golgi complex, which, what does Golgi do? What's one of the things that Golgi does? Ships?
Vesicles. And it's different.
So what's actually occurring in the little pancake? To me, it always looks like pancakes. Can't go wrong with pancakes.
Protein modification.
There's protein modifications occurring, and there's actually a pH gradient across the different stacks. I can't remember now if it goes from low to high or high to low. That part has slipped my mind since I'm not a cell biologist. Okay, we can also have viral or retroviral infections. In fact, one of them, like influenza, it's really cool because it has a huge conformational change. We're talking like tens, if not hundreds, of angstroms in length. It's got this long arm-looking thing that comes out and grabs the membrane that was whatever it's going to infect and pulls it in, and then releases its internal components within the cell. And of course, then we also have the fusion of sperm and egg at fertilization, and then in plants you have vacuoles, and then finally, mitosis. And so, all right, so here are the membrane receptors, and I actually go over this one multiple times. And we also have I don't know if it'll let me do it, but if you, on your own, I put a hyperlink in the picture itself to show, it's like sixth grade class, whatever, that does like a representative dance or something set to music of endocytosis and exocytosis through the Clathrin-mediated cascade. And so it's, given what it is, it's kind of cheesy, but it's still good. It still shows everything with respect to how Clathrin, the role that Clathrin plays. All right, but membrane receptors, these usually are oligomeric. Okay, so similar to the things that I showed a few slides back, we have multiple proteins coming in together. And almost always, you're going to have an external binding of whatever it's the receptor for, that's going to cause a conformational change. And that conformational change is what elicits the effect on the inside, internally of the cell. Okay, and we'll talk about, there are some exceptions, for example, some of the steroids and the sterols, which they don't elicit their effect until they are internalized into the nucleus. But when we do the cascades, the cell signaling pathways, we'll talk about this a lot. And so, for example, with respect to insulin and things like that. But here, the example that we're showing is LDL, which gets a lot of press nowadays. What is LDL? What is the LDL? What? Say it with some, what does LDL stand for? Low density lipoprotein. Okay, and so is it good or is it the bad cholesterol, even though technically it's not cholesterol? Is it good or bad cholesterol, your LDL versus your HDL? You think it's bad?
The media
You think it's
tells me
good?
bad.
What?
So the media tells me it's bad.
The media tells you it's bad, but it tastes so good, right? No, it is your bad, quote unquote, bad cholesterol, right? Because you always want to think high densities, because which has a higher density, protein or fat? Which is going to, because you just think about what floats on water, protein or fat? Fat. And so, right. And so that's why the more fattiness or more lipids that there are, the lower the density. So whereas the more proteinaceous it is, the higher the density. And so the LDL is your quote-unquote bad cholesterol. The HDL is your quote-unquote good cholesterol. What's also very important is actually the ratio, because, so for example, I use myself for an example a lot of times because I very rarely offend myself. But I'm so glad someone got the really lame joke. But no, my LDL is usually really, really good. It's a very good range, but my HDL, whatever with my metabolic pathway, my HDL is usually way too low. And so it makes my ratio out of whack. And we're not even going to get started on triglycerides until next semester. And that's actually a genetic thing. Same thing happens to my dad, my grandfather, and so on and so forth. But you want to make sure it's within a certain range for both your HDL and your LDL, and that the ratio of HDL to LDL is a certain amount, because otherwise you can have, what happens if it gets too high? Your LDL, for example, gets too high, or your HDL gets way too low. Why is it important to keep this in check?
Yeah, atherosclerosis.
You can get atherosclerosis. What else can happen? Which atherosclerosis can lead to?
Blood clots.
It could lead to the blood clot. It can lead to heart attack. Which, what's a medical term for heart attack?
Myocardial infarction.
One of the fun things to say, especially like in a Beavis Butthead kind of way, but myocardial infarction. Yes. There's another reason why. What else can go wrong? It has nothing to do with the heart, but if you're LDL, strokes, ischemia, just in general. So different types of ischemia or lack of oxygen. Okay, but what I wanted to talk about here is, and we actually go into much greater detail next semester when we go over the metabolic pathways for this, because it seems completely illogical the way that your body responds to this. At least I always thought it did. Of course, God didn't ask me whenever he was designing this, so that's the faith integration, I guess. But the LDL receptor is what carries the cholesterol into the blood. And so what happens is that whenever the LDL comes and binds to the receptor, it causes an initiation of clathrin or something similar to clathrin to come, and that's what internalizes it, and I'll show you in a moment. What happens is the clathrin, due to the conformational change, will start to come around and pinches that region around the receptor. And so it becomes more and more concave until it kind of just pinches it off. And I know something it's kind of crude, but it's a real world example. I don't know how many, or if any of you have ever been around this, but it's also a way that they, castrate bulls. Is because you just use rubber bands and stuff, and you just tighten it really tight, and with time it pinches it off. And so it's very similar to that. It's like clathrin. And so now you know. And then it's no longer a bull, but it's a steer. So, okay. But that's what happens with the LDL receptors. And so what happens is if you have too few LDL receptors, so one way to have atherosclerosis that has nothing to do with, well, I shouldn't say that. One way to have atherosclerosis is that you just don't transcribe and express enough of the LDL receptor. So if there's not enough receptors out there, then the LDL is going to accumulate somewhere, usually in your arteries. Okay? Or another way is if you have a faulty LDL receptor to where it doesn't get internalized. It doesn't do that conformational change. So that's a genetic way, predisposition to have atherosclerosis. But we're going to find out that there's a third way with respect to the receptors, and it does have to do with your diet. Like I said, on the PowerPoint, if you click on this, there's a movie where they've got these kids dressed like in hoodies and jumpsuits, and they're color-coded for the different membranes and everything. Oh, there's multiple myeloma. Okay, I mentioned clathrin, but the one that your book goes into the most detail actually has to do with caveolin, but they are very similar in the kind of functioning that they do. And so this is, the picture that we showed before was whenever the caveolin was a monomer, part of its raft. However, whenever something needs to be brought in and there's a conformational change that's occurring, you can have it dimerize. And if you notice, it does have a lipid anchors, but whenever it dimerizes, it becomes active. But it actually has a shape to it, like it causes that region of the membrane to be concave. So it forces the membrane around it to be kind of curved, and as more of them come around, it becomes more and more curved. And so that's what they're showing here, is more and more of the dimers start to form. And so you can imagine that since the lipids are relatively fluid, you have more of those proteins that are rigid, that are already concave, then it causes it to slowly pinch off. And this is just an electron microscope picture of that. You can see how they're forming in order for it to pinch it from the outside and it to become internalized.
So the caveolin is only do that when it's dimer?
Yes, that's whenever it's active. Because otherwise it's not, rigid enough to make it fully concave in that previous picture that I had of it. So this is just a blow up of that picture to show that you can see it. It looks even better down here than it does up on the screen. So at some point in time, you may want to look at that. But it's really cool the way it just pinches it, and then it gets internalized for it to go ahead and merge with the appropriate vesicle on the inside. I mean, not vesicle, but the appropriate organelle on the inside. Here we go. So some of the ways that you can cause, endocytosis or exocytosis, in particular, we're usually talking about endocytosis here, is if you already have a protein that is curved, which to me seems like the most obvious. If you have a curved protein kind of looks like that moon. Whenever it interacts with the membrane's going to curve around it as well. Okay, and so because of that, that's the lowest energy. So we could have some type of ionic interaction here if it's a peripheral protein. But just due to the lowest energy, it would actually take more energy for the protein to be flattened or for the membrane to not curve with it. So it's just energetically more favorable if the protein is already curved.
What would that be for?
The curvature of the protein to help initiate, like for example, endocytosis.
Well, for like endocytosis, wouldn't it be blocking like the where it would come in?
This is a blow up of it, but we're only saying that if you have enough of those around, then it's going to cause it. Or it could even be that the shape, maybe it's a morphological shape that something else will recognize it from the outside. So there are also other examples where, because of the curvature of the shape, let's say that you have something that starts to cause the membrane to curve, then that change in the shape will then cause a lot more of the individual monomers of proteins to come together, interact with each other, and that will cause a superstructure, and it will maintain that curvature, that shape of that membrane there. Okay, and so you have something else that starts it, that initiates it, then that activates and say, okay, more of these proteins coming and will accumulate, and you get enough of that, then it will help with the endocytosis, or later on we'll talk about exocytosis as well. And then finally, what if we have something, this would be similar to the caveolin that we talked about before, except this is just showing one, but we've got something that's got amphipathic helices or lipids that are stuck there, so it's stuck in the membrane. But the protein itself, once again, has the structure that's required that will force that membrane to bend to its will, so to speak. Are we on time? Okay, good. And so this is showing membrane fusion, and I know it's really small here, but I've got each of the steps blown up on subsequent slides, and so don't be alarmed if you can't read it. So, it's not letting me. This is the first step. Okay, so this is secretory vesicle, but it's not the only one that uses this, the snares and snaps. So sometimes in cell biology, they actually have, acronyms or names that make sense. And so this is just showing an example of like a neurotransmitter that's coming in and is wanting to merge for endocytosis. Okay, and what they have is the V snare, which is on the vesicle, and then the T snare, which is on the recipient. And just like, what would you expect like something called a snare to do?
Trap something.
Correct. And so the snares are going to interact with one another. Okay, that's what this is showing. So as they start to approach each other, the snares zip. They create these helical formations when they are interacting. And so that's how that they are complementary with respect to each other. And it brings the two membranes closer and closer together to where the snaps can help take over. And what do you suppose something called a snap is going to be doing? Snare traps it and snaps will snap it numbers. Okay. But what happens is as you get closer and closer and closer together, it's going to be more and more favorable to have these two membranes start to merge, because otherwise you're going to have phospholipid layer on top of phospholipid layer, and they would, in theory, would want to repel each other, right? And so it just, because you wouldn't want phosphates on top of phosphates, and so it's just easier for them to fuse together, and it's much more favorable then, okay, with the aid of these snaps.
How did they discover this?
I don't offhand remember. I mean, some of it was that they did through microscopy. Whenever they could see these two coming together, we still have a good 5 minutes, so they're really excited about problem stats. These two that's coming together, and they would take it, and you would be able to see it like in yeast and things like that, to where they would see them in different states of merging. And then I'm sure that they probably just did knockouts of different things to see the what genes compares to what and thing. But yep, they were smart in doing that. There's what's called the hemi or half fusion, and that's because the first lipid layer is going to connect with the next lipid layer. Fortunately, I've run out of the increase the actual slides because I didn't know how far I was going to be getting there. All right. And then we get the complete fusion to create like a pore, okay? So then that's when the neurotransmitter can be dumped into the inside of whatever the recipient cell is. Okay. So are there any questions? The snares, the V snares on the vesicle, the T snares on the recipient, those are what literally zip together. They do interact. They come close enough to snaps and take over to help fuse the two membranes together, and then you create this fusion pore, which is going to dump the contents on the inside of the recipient cell. So any questions or comments, concerns?